Our next speaker is Valentina Bonfatti, that I'm happy to introduce. Valentina is an associate professor at the Department of Comparative Biomedicine and Food Science at my former university, University of Padova. In her research, she focuses on the development of calibration equations to predict milk quality and meat traits to use for genetic selection. Her talk today will focus on the use of Bayesian regression models as alternative to partially square inferred prediction. So Valentina, you can go.
There we go. First off, I would like to start by thanking the organizers of the meeting for giving me the opportunity of giving this talk today. And I also want to thank the previous speaker, because for me, it was a really insightful morning. And I really appreciate the chance that I could join you for this meeting. I feel like I'm a little bit an outsider here because my background is not related to statistics or mathematics, but it's related to animal science. I should probably say that I am an outlier at this point. So the topic of today is the application. We're going to see some application of Bayesian regression models as a possible alternative to PLS. And during my career, years ago, I've been involved in a project with the aim of developing a number of calibration equations. We started by collecting roughly 1,300 milk samples out of a corresponding number of Simmental cows in northern Italy. The first aim of the project was to get to the prediction of a number of new traits, and we were interested at this point in working with milk composition and technological properties. But by adding population spectra to this data set, we wanted eventually to try to estimate breeding values, and especially to investigate how we could use or implement these equations into the current animal breeding programs. Of course, I'm not gonna go into detail of the genetic and genomic part today, but I'm just gonna focus on the development of the equations. For this project, we had the chance to work with 92 traits. We measured lactoferrin, protein composition by reverse phase HPLC. We got the measures of the 4 major minerals. Then we performed micro-cheese making experiments to get the cheese yield out of each individual sample. We measured the curd composition of that micro curds. We also got some measures of recovery rates, that is the proportion of fat, protein, or dry matter that we could recover in the curd as compared to their content in milk. We measured milk coagulation properties and pH, and also fatty acids. The reason why eventually we end up with so many traits in here is that because we wanted not just to investigate what was the potential of infrared spectroscopy to predict the concentration of fatty acid or protein fractions, but also to see if we were able to predict their relative concentrations, their relative amount in fat and in total protein. This is the distribution of the R-square of the equations that we got out of the project. And as you can see here, we have some traits with quite a good R-square. This results refers to the application of a PLS model. And the traits for which we could get a good prediction were the traits mostly related to total fat content and total protein content. So we have major groups of fatty acids, major fatty acids, casein, dry matter, cheese yield. You can also see that there is a whole bunch of traits with medium to high accuracy from 0.5 to 0.8. But we had also to work with traits that were, let's say, difficult to predict. This we have in the minor protein fractions and recovery rates that seems to be less related to the content of fat or protein and maybe more related to physical properties of milk. So the good thing about this data set is that we have a lot of traits to play with and all of the different R-square that we could get out of spectroscopy experiments. We thought about the application of Bayesian models because as geneticists, we are familiar with them because they are pretty common in genomic selection. In genomic selection, we want to estimate, or we try to estimate the genetic merit of animals by looking at their genotypes. And their genotypes are made of hundreds of thousands of different variables. So first we develop a reference population, imagine a few thousands of animals for which we have measured the phenotype we're interested in, and we also know their genotypes. And then we have to develop a prediction equation where we estimate the effect of each single genotype on the expression of our trait. By the application of this equation, we can then estimate on unknown samples what is their genetic merit, their breeding value. And if you think about this, this is very similar to what we do in infrared spectroscopy, where we have a training population, and then we need to predict some equation. And we try to apply that equation in practice to get to the most accurate predictions that we can. In genomic selection, Bayesian models are pretty common, especially if we assume that not all the genotypes have the same importance in the determination of the trait, but we have most of the genotypes with a very, very small effect and just some of them with maybe a bigger effect. And the theory behind Bayesian methods is that they use available knowledge about a parameter that is updated by using the data set we have. So this prior knowledge is called a prior distribution, and we have to set a sort of an expectation that we have around the parameter we are trying to estimate. So we need to set up this expectation without even looking at the data. And here the expectation is represented by this reddish distribution that represent the probability of having a parameter. In our model, when we do multiple regression, our parameters are the regression coefficients. So this would be the prior distribution of our regression coefficients. In a Bayesian framework, the second step is to estimate the likelihood distribution, here represented by the green distribution. The likelihood is the probability of observing the vector of our response variables given the parameter. Eventually, these two distributions are combined together to get to the posterior distribution, which is actually the probability of observing a given parameter given the set that we have. So this is the theory behind Bayesian methods, but we do have different options here in the way we set our priors. And in genetics and genomics, we call it a Bayesian alphabet, because we have different models that are different between one another depending on the priors that are assigned to the effects in beta, so to the regression coefficient. We can apply a Bayesian ridge regression where the prior for the effects in beta is a Gaussian distribution. This has the effect when combined with the likelihood to produce a shrinkage of the estimates to the same extent for all the estimates towards zero. We can have bias A, where the distribution is a scaled T, and it differs from the Gaussian because it has slightly thicker tails. So in this case, we have a more shrinkage towards the zero for the estimates that are close to zero. We also have other models like Bayes B and Bayes C, where the prior is a finite mixture distribution, in which we have a point mass at zero that is represented by this spike, and either a scale T slab for Bayes B or a Gaussian slab for Bayes C. And last one, we have Bayesian lasso, which has been already mentioned, but not in a Bayesian framework. And for this model, the prior is a double exponential. So we have these prior distributions for the models. Of course, we can somehow determine how we want that distribution to look like, our prior distribution. So we can set up different hyperparameters to shrink more or less the estimates. And somehow we can determine how we want to change our prior distribution. The reason why we decided to apply this or to test these methods is actually because there was a paper published in 2015 by a colleague of us, Alessandro Ferragina, And we have to give credit to him because he was the first one that decided to apply these Bayesian models to milk infrared spectroscopy. And they got really impressive results. They tested these models and compared some of them with PLS and MPLS on traits related to fatty acid composition and milk technological properties. And it got a massive increase in the R-squaring cross-validation. So I found this really, really interesting and potentially useful for my data set. So I decided to test these methods on my data. For model comparison, we had the spectra collected on a FOSS FT6000 for all our samples. I decided to remove the water regions, so eventually we ended up not using the entire spectra, 1060 variables, but I think around 870. Spectral variables were then standardized before the statistical analysis to a mean equal to 0 and variance of 1. And this is really important when the prior distribution is combined with the likelihood to get the posterior distribution in Bayesian models. Bayesian models were applied by using the package BGLR in the R software, and we compare them to a PLS where the number of components was set in order to minimize the prediction error. Models were tested in a 10-fold cross-validation where animals were randomly assigned either to the training set or to the testing set in respective of their farm of origin. So this means that we probably are a little bit overestimating the R-square of our models. And model comparison have been performed on the same training and testing sets for all the models. Let's say some results. Here you have the difference, the percentage difference in the R-square in cross-validation that we got for fatty acids expressed as grams per liter, so concentration of the different fatty acids. In the first column here, I have reported the R-square of the equations that we got from the PLS model. And in the other columns, you see actually the percentage difference of each Bayesian model compared to the PLS. And this table works like a traffic light. So if you see the white background, it means that the R-square in cross-validation did not change very much. And in this range, we have variations that goes from minus 0.5 to 0.5% R-square. So nothing really happening in here. With the light green, you see a little bit of improvement in the R-square in cross-validation for these models. Whereas for yellow or dark orange, you can see where actually for the traits for which the PLS outperformed Bayesian models. As you can see here, well, as a first glance, there are not many traits that actually benefited from the application of these models compared to PLS. PLS, as Tom said this morning, is hard to beat, but you can still see that there is a little bit of improvement in some of the traits, and especially for Bayes C. This is what we got when we expressed fatty acids as a percentage of fat. And here you see that for most of the traits, we got a little bit of improvement in the R-square from Bayesian methods, especially again with Bayes C. Here, I think the best results we got was an improvement of 2 points in the R-square for this trait here. So generally mild improvement. For proteins, we couldn't say the same. And if you look at these results, you can probably tell that for most of the traits, PLS actually worked better than Bayesian models. For some traits, like the unglycosylated capricaucin, we did have a massive difference in the R-square between the PLS and these models, and in particular for Bayesian ridge regression or for Bayesian lasso. For technological properties, we have a little bit of an improvement for coagulation time, the time that takes to reach a given firmness of the curd, and so on, but no real improvement when we apply the model to cheese yield or curd composition or recovery rates. Whereas, as you can see here in the last part of the table, we did have a worsening of the R-square in cross-validation when we tried this model on lactoferrin and minerals. So for these traits here, PLS seemed to work better. And in particular for minerals, we did get quite a few differences in the R-square and cross-validation, with 5 or 6 points also difference. Then I wanted to check also what happens to the root mean square error in prediction. And here things look a little bit better in the sense that for most traits, even when we look at the fatty acids expressed as concentrations, we did have an improvement in this parameter, with a difference that ranged between 2 and 4% for most of the traits. Again, Bayes C seems to be the best model in this case. And the same can be said also when we look at the fatty acid as a percentage of fat. Even in this case, for most of the traits, we got an improvement in the prediction error. Whereas for proteins, not really. It looks a little bit better than what we've seen for the R-square, but still we have some traits for which PLS performs better than Bayesian models and the other way around. So nothing really conclusive here. For technological properties, if we look at the prediction error, again, we can see that in most cases we have a slight reduction. Also for the cheese yield or curd composition at this point, we do have a few reductions also for the coagulation time. But if we look at lactoferrin and minerals, we didn't see any positive change even in the prediction error. So I decided to have a look at what happens on average, as I had 92 traits to work with. And this is the R-square in cross-validation that we got out of each Bayesian models if compared to the one given by the PLS across the 92 traits. So as you can see here, on average, Bayesian ridge regression worked a little bit worse than PLS, but we cannot say there is really a difference between the other models and PLS. For the error, we did observe a slight improvement, and especially with Bayes C. I want to point out now that actually all of these improvements seems to be really minor. There are minor changes in both the R-square and the prediction error. So I wanted to test how many traits had an improvement in both the R-square and the prediction error. And I set a threshold, let's call it like this, for significant. If the R-square did improve of at least 0.5%, and at the same time, the root mean square error decreased by 0.5%. And we only got 15 traits with these characteristics with Bayesian ridge regression, 20 traits with Bayes A, 28 with Bayes B, and 1/3 of the traits that responded positively, but in a limited way for Bayes C, and 25 for Bayesian lasso. If we increase that threshold for, let's call it the relevance of our results, to the traits that got a 1% increase in the R-square and at the same time a 1% decrease in the root mean square error, then these numbers really drop. And we only have 17 traits that benefited a little bit from the application of Bayes C. So I can summarize a little bit what we've seen so far by saying that we could not perform previous results. So when we actually applied these models to our dataset, we didn't have very exciting results. Bayesian models have the potential of reducing the error, especially for Bayes C, even though this is not always true because it is trait dependent. Bayesian models seem to perform better than PLS for some traits, but not for others. And in particular, we had generally good predictions for fatty acid composition, only minor improvements for milk technological properties, and these models were outperformed by PLS for protein profile, lactoferrin, and minerals. Also, if we really look at the number of traits benefiting from a relevant increase in the R-square or a relevant decrease of the root mean square error, the numbers are really, really low. So we can say that results are inconsistent and they are dataset specific. And we also observed this by looking at the 10-fold cross-validations, where we had some of these 10-folds that responded better with one model and maybe at the same time some other folds responded better to a different model. And certainly these models tend to be trait specific. So we will need to test which is the best model in each occasion. So I was trying to hypothesize why for some traits the Bayesian model seems to work better than PLS and why for some others they didn't. And I decided to look at the correlation between the R square of the PLS model with the change in the R square given by Bayesian models. So here my hypothesis, actually my question was, do the Bayesian models respond better if we have a better accuracy of the models in the first time? And this seems to be actually true partially, because there is a positive correlation between the R square of the PLS model and the increase in the R square that we have out of the Bayesian models. And we normally have this positive correlation with all of them. I think probably Bayesian A was the one with the less positive correlation. This is also confirmed if we look at the correlation between the root mean square error in prediction that we got originally from the PLS model and the difference in the error of prediction given by the Bayesian models compared to PLS. And also in this case, we have a correlation, and when the prediction error of the equation tends to be low, we also tend to have a reduction in that error given by the Bayesian models compared to PLS. And the last thing I have hypothesized was that maybe Bayesian models worked better if the models did not actually work for PLS. So every time PLS has a high number of components, we can hypothesize that it's not really efficient and it's kind of hard for that model to work. And what I found out is that there is, again, a correlation between the number of PLS components and the reduction in the root mean square error that we have out of Bayesian models. I just want to point out that these correlations are, of course, not very strong. So we can have, at the same time, a very high number of PLS components or a very low number of PLS components with two completely different behavior in terms of performances out of our Bayesian models. But in conclusion, we can say that Bayesian models tend to work a little bit better on traits that can be accurately predicted by PLS. That's not good news. Or traits that are predicted with a high number of PLS components. There is a large variability across traits in model performance. So what we can do is to test different models for the different traits. The benefits that we observed are generally limited, even though for some traits, actually, we may use some of these models because they perform better than PLS. And of course, more research is needed in this field. There is actually one practical application that we have that exploits Bayesian method in spectroscopy, and that we have applied to dry cure hams. The problem of dry cure hams is that the dry curing period, at least for the Parma ham, must be at least 12 months, where salt is the only preservative allowed. So the quality of the product comes from a certain level of dehydration. So we need to have a given level of dehydration to preserve the quality of the product. But at the same time, the weight loss at the end of curing due to dehydration is a loss of marketable product. So there is the interest of limiting as much as possible this loss. So our hypothesis was, can we select pigs for reducing ham weight losses, and how we do that by using spectroscopy. And for doing this, we had the chance of sampling 1,200 crossbred pigs. These pigs are produced in a seed testing program, so they are made especially to produce phenotypes to enable the selection of their sires, purebred. And we produced hams out of these crossbred pigs, and hams were cured for 12 months following the Parma ham product specification. What we did for this project was we went to the slaughterhouse 24 hours after slaughter, and we collect the spectra using this instrument. It's a portable, visible near-infrared spectrometer, a reading from 350 to 2500 nanometers. We took 5 spectra for each of the fresh sites by sampling the cross-section of the subcutaneous fat. So what we ended up having is something like this. We made use also of other predictors that were included in the model besides the spectral information. We did have for this body weight, back fat depth, carcass lean mean content. Then we had a bunch of traits that were evaluated by experts, round shape score, Marbley score. We also had subcutaneous fat depth measures taken by ultrasound. So we decided to combine all the information we had, and these are traits that are routinely taken for this group of pigs. So we decided to combine them also with the spectral information to try to predict what is going to be the weight loss at the end of dry curing. And we performed different models here. I tested 17 different models. I didn't represent it all here. Some of them used only the spectrum, some of them used other kind of information. The best model that we got used also, of course, the weight loss at first salting. That is the earliest available measure of weight loss that we have. But unfortunately, this measure also implied to have in place a individual traceability system for the hams. So we didn't want to use this information, and we selected this model to be the target for our selection experiment. So in this model, this is a Bayes-B model where we combined the average spectrum with these predictors. We had sex and carcass traits and fresh ham evaluation traits. And we got a final R-square invalidation that was 0.62. We did also compare this model with a random forest algorithm, but Bayesian regression actually outperformed the random forest algorithm. So we stick to this model here. And even though 62% R-square seems to be a little bit depressing, what we got when we estimated the genetic correlation between the actual weight loss after 12 months of seasoning and the predicted weight loss that we got out of this model, the genetic correlation was 0.9. This means that basically we can get the same accuracy for genetic selection if we select our pigs based on the infrared prediction as if we based our selection procedures on the actual weight loss. So since last year, this prediction model is now part of the selective program for this sire line, and the weight loss is now used as a new indicator trait in the breeding program, and this without any additional cost for the company. So I just wanted to close my my presentation by saying something that was not so depressing. So yeah, that's it. Thank you very much.
Thank you very much, Valentina, for your presentation. And there are a few questions on the chat, and then I have a few questions eventually too. So the first question is from Giovanna, and is, does it make sense to compare R-squared from the different methods when actually R-squared is very low for PLS?
Yeah, that's a good question. That comes to probably what I do. Because I tend to use, or I would like to use, infrared predictions in animal breeding programs. What really is important to me is to look eventually at the genetic correlation between the traits, between the actual and the predicted value. So I agree with you that when you have calibrations that do not really perform well, you also have a lot of variability around the estimate of the R-square or the prediction error. And yeah, I totally agree on that. But eventually we want to estimate something else. So yes.
Okay, yeah, I have a few questions as well. So in your last work, you mentioned that you considered also other like factors like sex or weight loss. How did you combine these informations with the spectra?
That's an interesting feature of the Bayesian regression models. You can actually combine different variables and set different priors at the same time for different variables. So for some variables, you can set, for example, a non-informative prior or a flat prior. And at that point is something like having a mixed model. Whereas for other variables at the same time, you can set a different prior. So this is possible with this Bayesian framework.
Okay, yeah, and also I also have response like about your results. So we also compared quite a lot in our studies. We compared different methods, and we saw exactly what you saw. Like the results are really data set dependent and trait dependent.
Yes.
Okay, another question is, are the bias A, B, C models slow to run? What inferential procedure I use and CMC?
It is a Gibbs sampler. No, it doesn't take much time. No, I wouldn't say it's very like it's not computationally intensive. I mean, if you compare this with random forests, I would say random forest is much more computational intensive than Bayesian methods.
Okay, I had also a question about if you look at the wavelengths selected after the application of Bayesian, because yeah, I presume that they do after the posterior, there are some wavelengths that are selected and maybe others that are shrunk to zero. So if you look if there was consistency in the across methods.
Because it wasn't actually one of our aims to look at the biological information underlying the models. We did not do that with the milk dataset, but I certainly did that with the dry cure hams dataset. So I actually looked at what were the wavelengths that were selected by the model. And especially because I didn't mention that, but we had different measures of the weight loss across the seasoning time. So we had very early measures, then some measures taken after 90 days or 5 months or at the end of seasoning. And I wanted to check if there was consistency between those sort of biological information. There was consistency. It was actually interesting to see how some of the variables were selected in the early stages of the seasoning, and some others were actually selected in later stages. And I think this comes from the fact that you may have different dehydration depending on the amount of water you start with. So it makes sense that not all the wavelengths are selected for any period of time.
Okay, yeah, thanks for replying to all these questions.